Great. So thank you so much for having me here today. I'm going to be talking about mobile applications for scaling mental health assessment. And a little bit of background on me is that I come from a different perspective from many of the other speakers today because I'm a clinical psychologist. So a lot of my research questions are motivated by my background in treating adults with anxiety and mood disorders. But I also wanted to start out by saying that while I'm currently at IU, Indiana University, a lot of the research that I'll be talking about today was done when I was a research postdoc in the Lab for Brain and Cognitive Health Technology at McLean Hospital. So this lab is run by Laura Germain. It's part of the Institute for Technology and Psychiatry at McLean Hospital. And this is where I did the mobile test that I'll be talking about. Okay, but before I do that, I wanted to give a little bit of background on the research that got me interested in asking these questions, which is based on this idea that across anxiety and mood disorders, we know that people have deficiencies in recognizing facial expressions of emotion. So a lot of the literature on depression and anxiety has shown that there's this negativity bias where people with depression tend to have difficulty with recognizing happy expressions, and they mischaracterize happiness as sadness or a neutral expression. So I got interested in this idea that is the more people misinterpret emotional faces, leading them to feel more depressed, and then they withdraw and experience decreased social support, and this really just leads to a vicious cycle of more depression. And is there a way that I could tap into that and potentially improve the deficit that people have in recognizing facial expression of emotion? So we also know, based on a wide variety of literature, that there are attentional biases in recognizing facial expression when people have depression. So a meta-analysis showed that if you are depressed, you're more likely to look away from positive expressions, and you're more likely to look towards negative or dysphoric stimuli. So I got interested in where people look when they're looking at faces, and I was reading a lot about regions of the face and got turned on to the idea of looking at oxytocin, which was a hot thing to be investigating in scientific research around the time I was doing this project. It is a drug that can be administered intranasally, so through a nasal spray. And it is associated with a wide variety of social, cognition, and emotional processes. And it's used a lot in control subjects, healthy controls, and it's been shown to improve the way that people detect emotional expression. Then it started being used in samples with autism disorder and showed improved emotion recognition. Then it started being used in samples with social anxiety. And I thought, well, depression and social anxiety have a lot of overlap. So could I administer oxytocin to make people with depression better able to recognize happiness? And I thought that this was really an important question, because if I could improve this deficit in recognizing happiness that we see in depression through a simple nasal spray, this could be a way that we could really target treating depression. It could improve social functioning and really help with people who had experienced a lot of recurrent episodes of depression as a way to enhance relapse prevention. So this is a research question that I was asking towards the end of graduate school, and I ended up miraculously publishing this study with null results, which was this really carefully designed study, a double-blind, placebo-controlled study of oxytocin's effect on emotion recognition in patients with anxiety and mood disorders. And after all of this, what I found was that oxytocin really did nothing in improving emotion recognition. It did nothing for visual attention, and it actually made anxious people slower in completing the task. So a big bust for oxytocin. But what this really got me interested in was what was wrong with the task itself. This emotion recognition task that I was using, which was a really standard paradigm in the field of emotion recognition, a morphed face task where a face starts neutral and transitions to a fully morphed emotional expression. I thought this was the best thing to use at the time. But I realized that there are a lot of measurement problems with it. So this is what really led me to thinking maybe it was the measurement problem in addition to the oxytocin that led to these weird findings. So these measurement problems of ceiling effects, particularly for happiness, are what make my results, I think, and all emotion recognition results really difficult to interpret. Even if you are extremely depressed or very, very anxious, it's likely that you're going to score 100% accurately in detecting happiness on standard emotion recognition paradigms because they are so easy. And additionally, most traditional emotion recognition tasks really conflate discriminability and response bias. So what I mean by that is that, for example, if you're judging a variety of faces and you've got fearful faces, sad faces, happy faces, and angry faces, you could make an error in detecting fear because you have difficulty discriminating fear. Or it could be just a bias towards identifying a face as not fearful. And even if a person has very high accuracy, let's say they have perfect accuracy for detecting anger, we don't know if that's just because they like to select anger for their response or they are truly really good at identifying anger. So these are the types of measurement questions that got me interested in using a larger sample and using mobile applications to really understand scaling these research questions. I wanted to use a really data-driven, well-powered approach, and I turned to using a non-clinical sample, the TestMyBrain sample, to see if I could map differences in how people change their ability to detect emotion across the lifespan. So this is a study that I used a different paradigm that was developed in the lab. It's called the Belmont Emotion Sensitivity Test, or BEST. I was able to recruit much more than my 60 subjects from the original study. I got 9,500 visitors to TestMyBrain who completed an emotion sensitivity task. They were mostly Caucasian, mostly female, but I had a huge span of age. So between ages 10, having kids do this study, up through ages 85, and a good range of age. You can see the histogram. So being able to use a different task allowed me to really determine how emotion sensitivity or subtle ability to detect changes in facial expression changed across the lifespan. So if you did this task, what you would see is for 1,000 milliseconds, two faces paired together flashing, and the faces would disappear and the question would pop up. To press 1 if the first face is more afraid, and to press 2 if the second face is more afraid. And this test actually, through multiple iterations, corrects a lot of the response bias questions in the early emotion recognition literature. So what we found here is pretty interesting. With anger in particular, we found using a segmented regression approach, which is really plotting out changes in slope, and they are called breakpoints, which is where the change occurs. We found an early breakpoint in the ability to detect anger around age 14. You can see that, and the confidence interval shaded in gray around it. And we found that the peak in anger sensitivity, or the ability to detect anger, was around age 29.6. Okay, so that's anger. Looking at fear, similar pattern with later breakpoints. So you see that with fear, the first breakpoint is around age 20, and the peak is actually later around age 33 and a half. Okay, so come to happiness, we see quite a different pattern. There's only one breakpoint, and it's early in life. So around age 21 and a half, we see a change and the peak in the ability to detect anger, and it stays pretty constant throughout the rest of the lifespan, which is different from the other emotions where we see a decline. Okay, so we can z-score them and look at them across. The major conclusions of this work were that anger sensitivity had the steepest slope. People learned to detect anger relative to other emotions much earlier. So we tied this to things like bullying and learning social cues at an age of adolescence that is developmentally appropriate. We also found that fear sensitivity peaked around age 34 and remained relatively high. So this was super interesting to me because I study anxiety disorders, and fear peaking around this age is really the time that anxiety and mood disorders are flourishing and stabilizing in adulthood. And then the last finding with regard to happiness was also interesting, which is that this happiness does not decline like the other emotion categories. It stays relatively preserved. And we linked this to the idea of socio-emotional selectivity theory of Carstensen, which is the idea that as you age, you tend to prefer positively valenced information to negatively valenced information. Okay, but what do these results actually mean? What was very great about this study was that in this huge sample, we were able to, in a non-clinical sample, we were able to map onto clinical risk periods for externalizing psychopathology, which are disorders like substance abuse and conduct, and internalizing psychopathology, which are the anxiety disorders, mood disorders, emotion regulation disorders. So that was cool. But overall, I realized if I want to continue this work in a clinical group, we need to map this onto clinical samples. So what I did next was look at this test, the BEST, the emotion sensitivity task that is an improvement psychometrically on others, to look at its effect on generalized anxiety disorder, which is the most common anxiety disorders, effect on ability to detect emotion. So again, across the lifespan, a population-based sample approach, I recruited over 7,000 people who completed the emotion sensitivity task that I mentioned, along with a measure of generalized anxiety disorder. So the GAD-7, generalized anxiety disorder 7 scale, is a scale where the higher you score, the more anxious you are in terms of generalized anxiety symptoms. And you can see that there is a clear association between the more anxious you are, the worse you are at detecting anger, the worse you are at detecting fear, and the worse you are at detecting happiness. So there's a connection there. And this study also allowed us to show what many findings have shown before, which is that there's a connection between age and gender and anxiety. And we were able to control for those things given the power and sample size that we had. So anxiety is known to decline across the lifespan, and females are known to report much higher levels of anxiety than males. And this was also shown in the TestMyBrain sample. Okay, so then I wanted to test this with depression. My original research question of if we improve the ability to detect happiness in depressed people, would their depression get better? And this was just published a couple weeks ago in Journal of Affective Disorders. I looked at, rather than anxiety severity, I looked at depression severity and the emotion sensitivity measure in, again, a very large sample of around 6,500 people. And here what you see is that the more depressed people are, depression severity was measured by the BDI, Beck Depression Inventory, along the x-axis. The more depressed you are, again, the worse you are at detecting emotion in faces, and that in general, people are okay, but not great at detecting fear. People are best at detecting anger, and happiness is in the middle. So again, most studies do not allow for this type of comparison because the tasks, being able to detect the face, they're not comparably easy or reliable. But in this test it is, so we can actually see these differences, which is exciting and cool. Okay, but how did I actually do this study, these series of studies, and what is TestMyBrain that I have mentioned? So TestMyBrain is a platform that was developed by my postdoc mentor, Laura Jermine, as a citizen science initiative to learn more about the mind and brain. So if you have heard of this website, it now has attracted over 2 million people taking the test. And how this website differs from the way that traditional psychology research is done has already been touched on by a lot of the presenters today. I know this actual picture has also been shown today by our first speaker, but we are really all familiar with the traditional brick and mortar research lab where you're brought in to do a highly controlled experiment in a very specific setting. But this method, we realize, sacrifices a lot of things. So you're sacrificing sample size, you're sacrificing generalizability and ecological validity for things like experimental control. So while experimental control was very important for initial studies, validation studies, we now know that we just need to expand, and I think that's what today's workshop is about. So the digital research lab is much more than the brick and mortar lab. It's using, and you might be thinking about the digital research lab as anything that can be done digitally on a phone, on an app, on a device. And it is that. It's anything that allows us to test scientific hypotheses about the mind and brain through a digital tool. But the digital research lab is much more than that because it's actually anybody who has access to the internet. So while not everybody has access to the internet, this is an outdated statistic that still shows a huge portion of the world has access to the internet. And we've seen a couple of figures like this already today too. Much of the world has access to the internet. So when we're thinking about the digital research lab, it's really anyone who can access the internet. And what we can measure is through a cognitive test pretty limitless. It's anything that can be measured with a desktop, laptop, tablet, or smartphone, Apple Watch. And people can do this research, participants can do this research at any time of day from anywhere in their house. And I'm going to talk about how. So our approach through TestMyBrain is to minimize barriers to participation. So, nobody wants to drive into a psychology lab to do a study. Here you can completely do it from the comfort of your own home at any time of day. That's minimizing barriers. The next approach, which I think is unique to TestMyBrain, is that, and maybe not totally unique, but was really exciting to me as I was getting involved, is that we share results immediately with the research participants. So whereas other studies you might do online, even research you're consenting to or not consenting to, you realize like you have to fill in your email address to get your results at some later date, and they're probably just trying to sign you up for something. Here with TestMyBrain, we give people their results immediately upon finishing the test in a way that's easy to understand, and I'll show you some slides on that. And then the third aspect of the approach is to respect a participant's time. So all of the tasks that are up on TestMyBrain at any given time are brief. And we do that on purpose, both because we know you lose people if a task is too long and boring, but also people are likely to do more things if they're brief, short tasks. They might do several studies instead of just one. And we are respectful of their time because we actually tell them how much time a study will take before they start it. So if you go to TestMyBrain now, you might not see this exact thing, but you'd see something like it. You'd see a variety of tests that you could click on. While some of them say 15 minutes, and I just said our tests are short, this is probably because there are multiple tests combined within a battery, but really the tests are designed to be short, simple, and kind of fun. So you can choose which ones you do. You go to the website. You could go to this website today and take some tests. And as I mentioned, we immediately share results with participants. So TestMyBrain got really popular around the time that people were interested in facial recognition, not from the emotion recognition perspective that I was interested in, but more on face blindness. So there are a lot of TestMyBrain tests that started with, can you recognize these famous people's faces, that have been like kind of shifted a little bit. So this is the example result page on a famous faces task. You'd see where you fall and where the average is. These are controlled for by age and sex. You also see where you fit compared to the other people. And this is a really easy way to interpret your results. We don't show like T-scores or other things like that. It's just plain and simple, your average. What we also measure on TestMyBrain is who is coming to the site. So what's interesting as we're studying engagement in science is that most people coming to TestMyBrain to do a task have never participated in research before. So this statistic might be a little bit outdated, but at the last time we looked, 68% of subjects on TestMyBrain had never done a research study before. They were simply coming to the site to learn more about themselves because they were interested in the brain, they were doing it for fun. You see a smaller portion of the sample was concerned that maybe they have cognitive deficits. We are very clear that this is not a diagnostic test. If you're really concerned about your performance, consult your doctor, consult your psychologist. But you see a wide range of reasons that people are coming to the site. So we're studying really why people are engaging in this. We're also studying the science of engagement. So in an early study on TestMyBrain, we were interested in recruiting people who had postpartum depression. And when the study was called Postpartum Depression, only about 33 people took this survey. Whereas when it was changed to Calling All Mothers, there was a huge spike in the number of people who took this test. So we're always looking at things like this on TestMyBrain, which is like, which are the most popular tests? Where are we seeing traffic? What is causing these spikes in traffic? Sometimes it's like a YouTube post of somebody doing some continuous performance task. There's a wide variety of reasons, but these are the types of things that are exciting, and we really want to see what is engaging people. And we take down tests that are not popular as long as we've met the quota of subjects we want. Okay, so you're probably wondering, I hope not, but is any of this valid? Many of the other speakers today have talked about the validity of web-based work, but in a really early validation study of the TestMyBrain platform, we found that results from web-based samples really don't differ in any systematic way from traditional lab samples. They compare in mean performance, performance variance, internal reliability. This is a little figure from that paper that actually shows the comparison of the traditional recruited lab-based work compared to the self-selected web-based sample, and we see very similar results. This is not the only study that's validated a lot of the TestMyBrain tests. While some researchers have taken specific tests to validate them, others, including Josh today, who gave a talk this morning, has looked at cognitive abilities across large samples and across the lifespan. We see converging results for internet-based work and traditional samples. I was also interested in getting experience with doing network analyses. These were kind of becoming really popular towards the end of my graduate training, and I wanted to see if we could replicate some network findings on TestMyBrain. So I worked with some researchers who are more interested in schizophrenia and schizotypy, which are schizophrenia-like symptoms. And we did replicate a prior network analysis using the TestMyBrain sample on a measure of schizotypy. So, time. You might be wondering about what are the pitfalls of an approach such as TestMyBrain, this citizen science initiative, and I do want to highlight a couple of them which you might be wondering about. One is the selection bias, and second is device variability. So when you are recruiting a self-selected sample through a citizen science initiative, you're going to get a range of people. And what's interesting about TestMyBrain is we tend to oversample adults and adolescents with an autism spectrum disorder diagnosis or autism symptoms. These are just people that might be more interested and curious about themselves, and they're taking tests online. So that is one thing that we acknowledge, is that our sample may not be totally representative of the population. The interesting question about device variability is something that we are just really trying to get a better handle on right now. We just got to revise and resubmit on this paper this week about looking at differences in user characteristics across devices. So when people come to TestMyBrain, we can tell how they are accessing the site, whether it's through a desktop, laptop, phone, or other sort of device. And we do see differences in demographic variables on the type of device people use. So if you are an older person who is more educated, you're more likely to use an iPad. And obviously iPhone, Apple users are smarter. I'm just kidding. But basically, there are differences in device variability, in demographic differences. There are also latency effects based on device, and I know that I want to save a couple minutes for questions, so I'm not going to totally explain all of this, but we see latency effects in Android users that are not due to the demographic differences that I highlighted on the last slide. They're really due to the device. So these are things that we really want to be controlling for in any measures of neurocognition going forward. We don't want to have differences that are due to which type of device a person is doing the task on. Okay, so I will end by talking about citizen science initiatives like TestMyBrain. Overall, the benefits do outweigh concerns about validity. People like getting feedback, and that makes them want to come to the website and do more tests. And immediate feedback is shown to enhance the retention rates of studies like this. So in my idea of reducing the burden of mental illness, what this is going to take through citizen science initiatives like TestMyBrain are really good tools that have been validated, open data, and collaboration. And I don't have time to talk about this last slide, but I will stop now to take questions and say thank you.
Okay, thank you, Lauren. Anybody have any questions? Again, you can use the chat or raise your hand in the Zoom participants window. So Lauren, I was wondering, can you tell us any more about anything else you've noticed in demographic differences across devices? Or is it just education and gender that you've looked at?
It's education and gender, which also relate to things like SES. We are not controlling for those types of things yet. It's really more about detecting these differences between the devices, which is just going to be important if we're moving forward with mobile cognitive assessments, to be able to say, well, is this due to the fact that they did the test on an iPad, or is it due to the fact that they're female, or is it due to, you know, some other reason? So we are not, that I'm aware of, and again, I'm a little bit removed from this work at the moment, but I think it's just device and some of those basic demographic variables like education and sex.
Okay, if there are no other questions, thank you again to Lauren. That was a really useful talk.